Hi, I'm Becca. I'm 10 years old and I want to know how cells were created.
I'm Beth Weaver, and I'm an associate professor in the Department of Cell and Regenerative Biology and the Department of Oncology at the University of Wisconsin-Madison. New cells are created from existing cells. One cell can make a copy of itself and form two new daughter cells. And the process that a cell goes through to form a new cell is called the cell cycle. There are two major tasks that have to happen every cell cycle. The first is that cells have to make an exact copy of their DNA. DNA is sort of like the instruction manual for a cell. It encodes genes for characteristics such as eye color and blood type. DNA is organized into chromosomes. Most bacteria have one circular chromosome. Animal cells, including human cells, have multiple linear chromosomes. And the chromosomes get replicated during the synthesis phase, or S phase, of the cell cycle. The second major task that has to happen during every cell cycle is for the replicated chromosomes to be organized and separated into opposite sides of the cell. And this happens during mitosis, or M phase, of the cell cycle. During mitosis, cells build a molecular machine, which is known as the mitotic spindle. And the mitotic spindle is composed of filaments that grow and shrink, which are known as microtubules. And microtubules connect to the chromosomes and organize them in the cell in such a way that they all line up in the middle of the cell. This is an artistic representation of that, and this is what it looks like in a real human cell, with the chromosomes in blue and the microtubules in red. After the chromosomes line up in the middle of the cell, they then separate to opposite sides of the cell, still connected to the microtubules. Then the cell elongates, which further separates those masses of chromosomes. And then the middle of the cell pinches off in a process known as cytokinesis to convert one cell into two cells. And that's how new cells are created.